Hello, everybody. I just want to thank the organizers for giving me this opportunity to present to you today. I am an MD PhD student at the University of Kentucky and feel thrilled to be able to present to you. As a trainee, it's an exciting opportunity to come here, especially a trainee from the medical field. I've spent the last 2 years in medical school and haven't gotten a lot of nutrition education, and what a great opportunity it is to be able to come with the leading experts of the field and to be able to learn more about that. And you know, it's been really inspirational and motivational to think about my future practice, what I might want to do and implement in my clinical practice and in collaborating with different nutritionists. I was talking to a lot of my collaborators and to some of you at lunch earlier this week, and I went to my primary care doctor on Monday, and I haven't had a primary care doctor in 7 years because I've been moving and I'm really healthy, knock on wood, and so I haven't needed to go to the doctor. But I was trying to, take control of my health and, maybe I'll look at my lipid profiles or my A1C levels or whatever, and he didn't do any labs on me because he said, you're really healthy, and even if I saw that you had high cholesterol, I wouldn't do anything about it. And even if I saw that you had a high A1C, I wouldn't do anything about it. And as I have been thinking, as I've been sitting down here and thinking about it, I'm like, well, maybe I would do something about it. If I had high cholesterol or high A1C, maybe I would want to know so that I can alter some of my lifestyle habits now so it's not so difficult for me later on in life after I'm, really ingrained in my ways. I don't need motivational change later on in life because I've already made changes now. And anyway, great to learn from you here. I want to share a little bit about my research story and what we've been looking at. I'm thrilled to be able to work with Dr. Ai-Ling at the University of Kentucky in the Sanders Brown Center on Aging, looking at Alzheimer's disease. Dr. Hossman-Cohen did a great job of talking about some of the background of Alzheimer's disease and the underlying biochemical processes that are going on there. There, of course, are plaques and tangles, which are built up in Alzheimer's disease, and the amyloid hypothesis has fueled drug discovery research for the past 50 plus years. We're trying to eliminate amyloid, and the problem is that we have done that in mouse models several times, and we bring it back into the human population, and there are either huge side effects or it's just simply not effective. And we believe in our lab that there are underlying metabolic features, that there are errors of metabolism going on that are potentially driving the amyloid, the Alzheimer's disease processes. This makes sense. APOE is the biggest gene which is altered in Alzheimer's disease. The APOE4 genotype is, as we've heard, correlated with threefold increased risk of developing Alzheimer's disease. And there's a great Nature review paper that talks about how APOE is involved in neuron survival, microtubule stability, synaptic plasticity, regeneration. The brain is full of lipids, and it makes sense that the APOE gene is so involved in Alzheimer's disease and neurodegenerative processes. In order to look beyond the amyloid hypothesis, we decided to interrogate the biochemical profiles of post-mortem human brain tissue in Alzheimer's disease. We're very fortunate at the University of Kentucky to have the longest funded Alzheimer's disease center in the country, which means that we have access to incredible resources of data to look at how Alzheimer's disease has affected people over their lifetimes. The members of the community are extremely giving, and that they'll come year after year after year for cognitive and memory testing, and they'll even get a lumbar puncture year after year so that we can collect CSF. And all of these people have donated their brains to the Sanders Brown Center on Aging, and I feel so lucky and incredibly grateful to be able to use that human brain tissue to look at some of the processes that are underlying this terrible disease process. So we looked at 24 different human brain tissue samples, 6 APOE3 controls, 6 APOE3 Alzheimer's disease, 6 APOE4 controls, and 6 APOE4 Alzheimer's disease. We looked to see if there were any major differences between these groups based on their clinical profile, to see Okay, was their age at death, their sex, their education, their hypertension status, their stroke status, their cancer status different amongst all the groups? And the only two differences we could see amongst all the groups was in Braak staging and their last MMSE. Braak staging is a neuropathological report to show how many plaques and tangles are in the areas of the brain to show severity. We would expect to see differences between control groups and Alzheimer's groups. And MMSE is like the MoCA or the other neurocognitive testing, which is used to monitor how far progressed Alzheimer's disease is. We would also expect to see differences between control and Alzheimer's disease brains. We interrogated 651 different biochemicals to see if there were metabolic differences between these groups. I'm going to focus on this group in this talk right here. These are all APOE4 genotype. And we're going to be comparing the Alzheimer's disease versus control brains. So they're all APOE4 genotype, and we're comparing Alzheimer's disease versus control brains. We saw that 44 biochemicals were upregulated, and 29 biochemicals were downregulated in that. So this has implications. If you get your genotype back and you see that you have APOE4, it is not deterministic. It's not going to tell you that you're for sure going to get Alzheimer's disease. But we saw that there were differences in metabolism between those that did develop Alzheimer's disease and those that didn't. And it's interesting to us to look at those to see if there are implications for how we could potentially change those in the future if somebody does get that back in their genotype. So we began to see some patterns that emerged as we looked at the data. When you look at 651 different biochemicals, there's all kinds of data that you get back. But we saw a lot of patterns that began to emerge, and the patterns that we saw were that Alzheimer's disease in the APOE4 genotype was related to metabolic syndrome, to mitochondrial dysfunction, and to kidney dysfunction. And I want to go through each of those individual metabolites now. In metabolic syndrome, we saw decreases in betaine, increases in alanine, and decreases in phosphatidylcholine. Betaine, it's consistent to see decreases of betaine in metabolic syndrome. It's also related to insulin resistance, prediabetes. We talked about betaine earlier on in this conference, so I'm not going to talk a lot more about it, but alanine is involved in the glucose-alanine cycle. If your body needs to ever break down muscles for energy use, it can then transport it to the liver for glucose uptake. Your alanine levels in your blood tend to parallel your blood sugar levels. So you have higher alanine levels that correlates with higher blood sugar levels, which is a risk for metabolic syndrome. And in addition, we had decreases in phosphatidylcholine, which is an important lipid bilayer component. Phosphatidylcholine is also very important in metabolism and signaling. In addition, we saw metabolites which were altered relating to mitochondrial dysfunction, cysteinoglycine and succinate. Cysteinoglycine is a breakdown product of glutathione. Glutathione is, of course, really important if you do have mitochondrial dysfunction in mitigating the reactive oxygen species that occur as a result of that. And if you are consistently breaking down your glutathione into cysteinoglycine, you're not having as much of that available to mitigate those processes. Additionally, with succinate, it's interesting, succinate dehydrogenase is the enzyme which is responsible for converting succinate into fumarate. Succinate dehydrogenase is important for the health of the mitochondria. And so it's natural if that is altered, there are decreased levels of it, then you'll have increased levels of succinate. We see 5-fold increases of succinate in Alzheimer's versus control patients in the APOE4 genotype. And lastly, talking about kidney dysfunction, we saw alterations in glycerylphosphocholine, myoinositol, and arginine. Glycerylphosphocholine and myoinositol are both consistently seen as increase in kidneys which are under stress. So your kidneys will produce these when they're under stress, and arginine is related to the urea cycle, which is also in kidney dysfunction, you'll have increases of arginine. So after seeing all of these different patterns, I went back to the literature to see, are these consistent alterations, things that are seen in other people's research, and indeed they are. There are many, many, many papers talking about kidney dysfunction in Alzheimer's disease, mitochondrial dysfunction in Alzheimer's disease, and metabolic syndrome in Alzheimer's disease. And I even found this one paper here. This comes from a 2014 review, which talks about all three of those. Leptin, of course, is involved in mitochondrial biogenesis. Angiotensin II is involved in the kidney health, and insulin involved in metabolic syndrome. These are all biomolecules which were found as altered in Alzheimer's disease in this review paper. So this is really exciting to see that there are metabolism differences. So even if you do have the APOE4 genotype, it's exciting to see that there are metabolism differences. It doesn't prove that kidney dysfunction leads to Alzheimer's. It doesn't prove that metabolic syndrome leads to Alzheimer's, but it suggests that if you do get this in your genotype, then you can think, wow, I need to be really careful about my kidney health. I need to be really careful about avoiding metabolic syndrome. I need to be really careful about my mitochondrial health. And perhaps I can work with a nutritionist or with another licensed healthcare professional in order to really optimize health in these areas. And maybe that will help me to decrease the risk of incurring cognitive decline later on in life and incurring that Alzheimer's pathology. So I just want to thank everybody that made this research possible. Again, I want to thank my mentor, Dr. Ai-Ling, who has been an incredible mentor. If anybody is struggling with mentoring, go talk to her. She's an incredible mentor. I'm so grateful for the Sanders Brown Center on Aging for providing the resources for this, the University of Kentucky MD PhD program, and for Metabolon for making this analysis possible. Thank you.